Cool. Think we're recording. Hopefully you can hear me, because I'm not doing this over again. So, what's up, Dr. Ray, and all the other hundreds of thousands of people on my famous Instagram page. Welcome to the show. Today, we are going to dive into why you aren't good enough. Apparently, Dr. Ray, that was what you were trying to get me to feel like, as this old man stood on the screen telling me how my DNA isn't enough. So I guess today we'll be talking a bit about my worst moments and why I'm such a failure. No, I'm just kidding. Today we'll be talking about why your DNA, my DNA, and apparently this stupid, glitchy background on Zoom isn't enough. We're taking a deep dive into epigenetics and the important role it plays in life, and why I need to call Zoom or PowerPoint or Microsoft to get a grip on what's happening here with this glitchy background. And hopefully, for comedic relief, it's not glitchy when this plays back. So I just look like an idiot up here talking about a glitchy background that's not actually glitchy, that looks cool with the DNA molecules in the background. But nonetheless, if it is glitchy, I hope you're laughing at me right now. Um, Honestly though, I did really feel like I wasn't good enough when I was going through this. I was like, damn, like my DNA isn't good enough. This is why I'm a failure. This is why I'm no good. I cried a few nights in a row. Could barely get up this morning to do this talk, but I got to do it for the fans. Anyways, why your DNA isn't enough, but is still very important. All right, so let's get into this, okay? What is DNA? Why is this old man on this screen telling me that it's not good enough, but also telling me that it is important and all of these things? I got kind of confused. So let's kind of just start with the baseline. What is DNA? Well, first and foremost, DNA is the foundation of our genetic understanding. It's the makeup of who we are as a species, genetically. It encodes how tall I am. It encodes the color of my skin. It encodes the texture of my hair, the color of my eyes, my facial structure. It encodes the way that my heart is situated in my body. It encodes everything that makes up who I am. But one very important thing about DNA is that it is a fixed molecule, right? And that is something we'll go into later. It kind of feeds into this process of understanding, like I mentioned earlier, epigenetics and understanding the difference between fixed and dynamic. And so it's important to recognize now that DNA is a fixed molecule. And like I said, it encodes the hardware of who we are, right? It encodes the basis of who we are as people. And I did this kind of thought experiment when I was doing this slide, and I wanted to do it here. I didn't want to think through it while I was actually doing the slide because I wanted it to be like a live thought process. But like, what would happen, honestly? And Dr. Ray, you're a geneticist, you might know. What would happen if right now, sitting here in this chair, all of the DNA in my cells disappeared? Like it just evaporated. How long would it take me to die? Would it be, I mean, because it can't be instantaneously, right? Because like obviously you have your DNA, your RNA, and your proteins, right? So you have the proteins necessary at each moment in life to to basically live and survive. And if you need more of them, obviously your DNA turns into RNA, RNA turns to protein, and you make more of those proteins. But how often are those essential proteins how often do those essential proteins need to be regenerated? So like I was just thinking like how long would it take for me to die if my DNA just evaporated right now and everything else stayed the same? Everything else in my cells stayed the same. Just the DNA in the nucleus completely evaporated. You let me know when you grade this. Okay. Lastly, DNA, it gives us a baseline or a starting point for epigeneticists to begin exploration. right? It gives epigeneticists a space to start and begin looking into how might this fixed molecule play a role in the way that epigenetics kind of manifests itself in the world. And now, Dr. Ray, I know you aren't thinking this, but to all my other fans out there watching this video, I want to make sure that I'm being personable, being humble, and I'm not forgetting about you. And so I know exactly what you're thinking. What the is an epigeneticist? Now, because I'm a good man, I'm not going to cuss on this video. But what I will say is I'm thinking right there with you. What the is an epigeneticist? Well, I got the answer for you. An epigeneticist, okay, studies factors on top of or in addition to genetics, right? So epigenetics is the study of factors that are on top of genetics. So you can think of this as a study of the things that are on top of or that happen to DNA, right? And so epigeneticists, basically what they do is they go in, they look at DNA, and they have now identified, oh, well, there's endogenous or exogenous rather changes that happen to the DNA that manipulate, say, a phenotype. And so those epigeneticists study the process of the factors that change DNA, that don't change DNA, because DNA is a fixed molecule. But epigeneticists are study the factors that kind of manipulate or add to DNA to create us as individuals. And so the effects of epigenetics, factors have relationships to a bunch of different biological elements, right? We see epigenetics in developmental biology, right? We see, epigenetics in disease, in the environment, and in individual variation, right? And so we'll get into these in just a second, but just to kind of round that out for everyone to take a deep breath, we now know what DNA is, we now know what epigenetics is, and we now know the stunning and brilliance of what these people, epigeneticists, are and what they do. Now, let's understand the relationship between epigenetics and a few of the things that I talked about or that I mentioned earlier. So first, epigenetics and development, right? But there's two main points here, and I'm sorry these slides are boring, but I'm going to do most of the talking here. I didn't want to put too much, words on the slide, so I'm going to take on the load of the creative expression here in the next few slides. But basically, epigenetics and development have an interesting relationship in two ways, right? As we experience the world, it has a way of creating epigenetic marks on our DNA that then induce or encourage these sections of DNA to express or not express. And these things will be talked about in a little while. But let's just say, for example, we can kind of go the creation of the epigenome through experience and young brains and genetic potential. Those 2 points are kind of meshed together, and here's how. As we move through the world from a newborn to an old person that's about to die, through the entirety of our life, we are developing, right? Obviously, we develop more in our younger ages, but we're developing over time. And what happens is our experiences with the world tend to create our epigenome, right? So we start off with a set, strict, fixed DNA, right? That's immediately in our embryo. That's our DNA right there. We have it immediately and it doesn't change. However, like I said earlier, what happens is this epigenome gets created by adding marks or things on top of your DNA. And that's how your genetic potential, right, is expressed. Our young brains experience the world. The world then gives back to us impulses, signals, or whatever you want to call it, that goes back into our genome, creates marks on our DNA, and thus the creation of the epigenome arises. Okay. And so the epigenome is something like marked up DNA. And we'll get into what marked up means soon, but we can just kind of use that framework. Epigenetics and disease, right? We have external factors like the way a mother eats or drinks or acts or the relationship she's in and how those things affect the embryo epigenetically can induce birth defects, or even later on in life epigenetic mutations can induce cancer, which is something that we'll talk about extensively further, so I'll kind of leave that there for now. Epigenetics and any environment. This is a really big one, especially right now with climate change and things of the like. But epigenetics and environment is going to include things like pollution, and that can be air pollution, right? With all the smog and the smoke, we can think of places like Los Angeles, or even places overseas like India that are really bad with air pollution. We can think of water pollution, right? In our oceans and our rivers, we can think of microplastics, and oil spills, right? These things all play a role in the way that our genome reacts to the world, right? Reacts to the environment. And also radiation, right? This goes into, a little bit more of climate change, and understanding, holes in the ozone layer that protect us from that solar radiation, and how even something like the environment, right, might induce epigenetic changes in your genome, and then also interplay with how and also trigger disease like cancer, right? So we can see these epigenetic kind of these different factors or these different categories, or relationships epigenetics has with different things can actually relate together in terms of the environment and, say, disease. And lastly, we see epigenetics and individual variation, right? This is one that most people are understanding of and recognize, is that epigenetics is affected by your diet, your exercise, your level of education or your quality of education, and your relationships, right? As we talked about earlier in development, right? We all develop as children, and we all have different types of parenting. We all have different types of, friendships, different schools we go to, different experiences that we have, all of these things, right? What we eat for snack time. Do we run around at the playground during recess, or do we sit inside and draw, right? Or do we go to a private school, or do we go to a public school? All of these things affect our epigenome, by inducing different types of experiences with different levels of kind of severity. And then our genome gets marked up accordingly. And so this is the essence of what makes us individuals. And I don't talk about this in this specific talk, but there are studies that are done that look at identical twins that were raised in two different environments, where their genome and their genetics are identical, but their epigenetics are what show them to be completely different. And so if two identical twins can grow up to be different, you better believe that someone you don't know or someone across the globe, even if they have similar DNA, can be vastly different than you in terms of their interaction with the environment and how epigenetics plays a role in that. So basically, big picture epigenetics is basically, our DNA is the foundation, but the factors that induce change or variation are the product of epigenetics. And so as we see here, we have DNA on the left side of the screen as the tree, right? A tree is a tree is a tree, right? And what happens is the tree most of the time looks like this, right? But as the environment changes, as the tree starts to experience things, it begins to change, right? As the DNA gets input from the world and the environment, it begins to change. And so, maybe sometimes the green leaves might turn red, or maybe sometimes the environment will induce a shedding of the leaves. This can be kind of assimilated with or thought of like the way that our DNA gets methylated and acetylated, or demethylated and deacetylated. This is something we'll talk about in depth in a few moments, but I want to make that connection. We have our DNA. the environment gives us a signal, and our DNA then gets added to or taken away from externally, right? As a result of that. And so it's the same DNA, just different things on top of it. Let's just say it that way. And so, okay, 15 minutes in, let's take a breath because we're about to start moving here. And so up next, what we're going to talk about is bridging the gap between tree leaves, changing colors, like, oh my God, and epigenetics at the DNA level. First, I need to say, I am very, very sorry. I am very, very sorry for the biochemistry that is about to come up. I know you're a geneticist, Dr. Ray. I know, I know. No one likes biochem. I know. Hence the meme. But I just do need to say sorry and sorry again because this is a genetics class. I know you probably don't like biochem. Neither do I. But here we are. I really wish I could take a 30-minute break right now, every 5 minutes, but I can't. All right, let's get this done. So, how can you change gene expression? What does that mean? I thought DNA was fixed. I thought genes were fixed. What does that mean to change gene expression? Well, you can change gene expression by adding or taking away these things called methyl groups or acetyl groups from DNA and histone. So if you modify histones in DNA, what happens is you can actually change the way that genes are expressed. So DNA modification, right? What DNA is essentially, as you can see in this, or over here, maybe it's down here, in this graphic here, is that DNA is condensed and coiled around what's called nucleosomes and are methylated by DNA methyltransferases. Histones, on the other hand, can be manipulated as, they are subunits of what's called nucleosomes, and they modulate DNA spacing, right? And they are manipulated by histone acetyltransferases. Now, as we understand this, chemical manipulation of DNA or histones can actually increase or decrease gene expression. And so the mystery of methylation and acetylation basically comes down to two things: histone acetylation and DNA methylation. And I must note, I say mystery because I don't know why it took me so long to understand and wrap my brain around the effects of methylation and acetylation. Um, I had to take, I literally did this. I was online and I'm looking, okay, what does acetylation do? What does methylation do? And some papers in Nature were saying, methylation reduces expression. Some are saying it increases expression. Some are saying acetylation does this and that and the other. I got overwhelmed. So what did I do? I grabbed my good old Campbell biology book, flipped it open to DNA and gene expression, found methylation and acetylation, and I used my textbook for the first time in 37 years. Back when I was at UC Berkeley in 83, I used to be in textbooks all day, every day. This used to be my thing. I used to love to read. I was a bookworm in textbooks. Now I got these computers and these iPads and whatever. And now I'm online and it's all overwhelming. I had to get back to my roots. Went back to 83, UC Berkeley, you know what I'm saying, the library, where I'm in there, I'm reading words on paper. Dr. Ray, I know you understand. Everybody else that's seeing this are going to be the younger generation. They're not going to say, what is a textbook? What are you talking about? What are you talking about? A textbook? What is this? Me and you, we right here, man. We right here. Anyways, but what I found from my textbook was that histone acetylation actually promotes euchromatin state. The euchromatin state is what allows for increased transcription and gene activation. And this is very important because it plays a role in the ability for scientists to go in and manipulate different genes in experiments. And so what happens is, specifically, there is an acetyl group added to the lysine amino acid of the histone tail. And now what scientists can do is that they can go in and they can acetylate this lysine amino acid on a histone tail and end up activating genes that they want to see expressed in experiments, right? So you can see this as like an activated, gene state. However, whereas DNA methylation promotes a heterochromatin state. This is where you have decreased transcription and you have the silencing of a gene, and basically the histone tail is methylated at the cytosine base. And this is where you see in science and in experiments, most of the time, scientists are going to knock out or silence genes. And this is one of the mechanisms that allows scientists to go in and knock out or knock down or silence genes by methylating the histone tail that the gene is associated with, and thus removing the gene and seeing the effects of that removal or of that knockout. You most often see silenced or knockout genes in the way of this DNA methylation. It's rare that you find kind of activated genes or knock up or knock in genes. So that's just one thing to kind of note. But again, acetylation is going to increase transcription and activate the genes, and methylation is going to decrease transcription and silence genes. Shout out to handy dandy textbook. Now, talking about science, right, we want to move into how epigenetics kind of came to life in some real sense. And this is a image of Dr. Paul Mark's results of a study of his cancer therapy, right? And this drug called Zolinza, I think, was the first FDA-approved epienzyme inhibitor. And I know that sounds like, what are we talking about here? All this is showing is that on the left side, this patient has tumors highlighted by the red arrows. And after taking this drug that was that targeted epigenetic proteins that induced tumors, basically what happened is that after treatment, those tumors started to shrink. And so this drug targeted these proteins, right? And ultimately induced tumor regression. And so epigenetic proteins now became kind of this main target for cancer drugs. In 2006, there's this experiment done. It showed the regression of tumors, and all of a sudden it showed that the machinery associated with epigenetics played a big role in the ability to kind of cure cancer, if you will. And so, or at least cure tumors, and was thus kind of denoted as, okay, maybe we can attack cancer in the same way. Get a sip of water here. All right. Now, from here, people started looking at the other kind of parts of epigenetics, right? So first, we looked at disease, that last slide, right? Remember we talked earlier about epigenetics and disease, right? That last slide was looking at disease. However, other experiments and other scientists wanted to also look at epigenetics and the environment, or epigenetics and behavior or development. And so these are two really important experiments that highlight the foundations of what epigenetics are. And so first we have the diet experiment. This is where genetically identical mice, and only differed by mother's diet, right? By the mother's diet, produced children of varied health. So obviously, to the right, the mice are a bit more chunky, and to the, or I don't know, yeah, to your, no, to your left on the screen, on the left. Here we go. I'm here talking about epigenetics. I don't even know my left and right. Can you believe that? Anyways, on the left, the chunkier mouse kind of is indicative of the mother mice eating more, and to the right is indicative of the mother mice eating less. And so what we see is that if you're identical genetically, and even just your diet changes the way your offspring kind of develop. And also there's a good mom versus bad mom experiment where maternal behavior is shown to be passed on to offspring, right? So we see that mothers with good behavior, mothers that lick their pups a lot, mothers that cuddle their pups a lot, mothers that kind of feed them properly and on schedule, show to have offspring that actually do those same behaviors. And so here, basically what this is showing is that in the first experiment, your diet even the maternal diet has an effect on the offspring, but also the maternal behavior has an effect on the offspring behavior. And so ultimately, behavior can induce epigenetic marks that yield different phenotypes across offspring. There's another, benefit of understanding epigenetics, and that is going back to this idea of the histone code hypothesis. We talked about histones earlier, right? We know that we have our DNA in chromatin, right? When it condenses down in a chromatin, that chromatin wraps around what's called a nucleosome, and the parts of that nucleosome are called histones. And the histones, like I mentioned earlier, are what gets methylated or acetylated, which then causes increased transcription or decreased transcription. Now, what's so cool about this is that the identification of certain histones allowed scientists to create a code. And this code basically allows scientists to identify parts of the genome that are open and actively expressing, and parts of the genome that are closed and not actively expressing. Right. And so here, as we can see in this top green box, we see histones like H3K4ME1, H3K27AC, H3KME3, and H3K36ME3 that are more active, that are upregulated, that are on, that are in this euchromatin state that are allowing for transcription. However, in the bottom box in the red, this H3K27ME3 and this H3K9ME3 are histones that are more often not overly expressed, not in that euchromatin state, but rather in that heterochromatin state, and not often expressed. And so by looking at these different experiments, right, we can understand that scientists were able to not only look into how epigenetics relates to disease and how epigenetics relates to behavior, but also kind of create this code of the human genome and trying to understand which histones do what. And it ultimately gave them a map of histones to target based on the goal of their experiment, whether they wanted to knock in, knock out, upregulate, or downregulate different types of genes. So now we can get into something very specific, which is pediatric cancers and how they point to histone mutations. We talked about the histone code hypothesis and looking at specific histones. And what's very, very interesting is that actually a lot of pediatric cancers point specifically to histone mutations as the cause or as the kind of starting point for where these pediatric cancers come from. And one of these cancers is called diffused intrinsic pons glioma. And this is a very, very fatal, universally fatal tumor that usually is produced in children, and once diagnosed only gives them about a year and a half to live. And what happens here is that the lysine mutates on the histone tail, right? As seen in this graphic here. The lysine mutates to a methionine amino acid at histone 3 tail. Now, this is detrimental to the function of the histone because the genetic mutation impacts the epigenetic machinery that is used for expression of that specific gene. And so what happens here is that the lysine turns to the methionine. The machinery that is used to methylate or acetylate this histone 3 tail can no longer read the histone tail. And all of a sudden what happens is now you can get this overexpression of cells. And now this tumor, which is just a bunch of cells dividing uncontrollably in the pons, which is in the inside of your brain. If you think of the brainstem, you have CMP, so you have your cortex, medulla oblongata, and then your pons, CMP, LMD. So you have your cortex, medulla oblongata, pons, lower CMP, MLD, right? Which is your medulla oblongata. Oh, no. Your cortex, your midbrain, your pons, your medulla oblongata, your lower medulla oblongata, LMLD. and then your dorsal root. And so the pons, sorry, it took me so long to remember that. I haven't gone over it in a few months. But your pons is actually right in the middle here of your brain, your cortex, your midbrain, your pons. And so there's no surgical way of getting in and removing that tumor. And so now you have children with these tumors in their pons area, and it's just fatal. It's just fatal. It's just real bad. And so this kind of mutation was now noted as a oncohistone mutation, which is basically referring to onco being cancer, histone being histone, and a mutation on a histone that is kind of cancer causing. So oncohistone mutation is kind of a buzzword, but nonetheless, that's kind of what it's called. So diffuse intrinsic pons glioma is basically one of probably the many very fatal diseases that are caused through the effect or manipulation of or detriment to epigenetic machinery. Now, to conclude, we've been able to kind of talk about what is DNA, the foundations of DNA, how DNA plays a role in epigenetics, how epigenetics chooses to or goes about kind of adding to that DNA with the acetylation and the methylation, how the acetylation and the methylation plays out in real time in relationship to development, disease, environment, and individual variation. We've talked a bit about the first clinical application of epigenetics and how targeting epigenetic proteins and machinery actually showed promise for treating cancer. And we also looked at a few different things that scientists utilize when it comes to, the field of epigenetics. And so I want to close with the future of epigenetics and understanding where this field might be going. So first, right, this is a very promising thing here. The future is looking into detoxification of oncohistone mutations to develop new therapeutic approaches for cancer. Now, mu is actually a Greek symbol, and it's definitely not supposed to be there. But I am 33 minutes into my presentation, and I refuse to start over. So please forgive me. As I said earlier, I know I'm not good enough. But hopefully one day the universe will conspire to induce positive epigenetic marks on my DNA so that I can be successful and I can be good enough. Also, the future of epigenetics will look into developing a deeper understanding of the methionine amino acid, since this K to M, or this lysine to methionine mutation, is causing so much trouble, right? You have 20 essential amino acids, and you have this one switch, right? There was an experiment done by Dr. Alice's post-doc fellow that looked into understanding if you turn the lysine into one of all of the amino acids, what would happen? And methionine and isoleucine were the only two that had any effect. And so it's interesting because you have non-polar amino acids, you have polar amino acids, you have acidic amino acids, basic amino acids, and so on, aromatic amino acids. And so to see that just these two specifically have changes and cause so much trouble is really interesting. So scientists want to develop a deeper understanding of that as well. And lastly, epigenetics is very, very broad. And so the field wants to dive a bit deeper into epigenetic effects on neurology, fibrosis, cardiology, and ultimately immunology. And I'm sure many, many, many more uh, topics and fields. And so that's 35 minutes, 15 over. I hope I did enough to make you feel like I am good enough, Dr. Ray. To all the hundreds of thousands of people out there, go pick up a textbook. I promise you it's better than online. Back in 83, that's all we used, and here we are. So yeah, I hope you enjoyed my talk about epigenetics. I had a great semester with you learning genetics. I really did not like genetics before taking your class because it was just Punnett squares and, peas and I just didn't get it. I didn't understand it. It wasn't something that I was interested in, but all jokes aside, I really, really did enjoy learning about all the different things that go into genetics, right? Understanding Punnett squares and their importance of understanding genetic, inheritance, looking into genetic variance and how that plays a role in how different genes are passed on, even down to looking at the bacterial, transformation, transduction, and conjugation, right? Understanding the way that bacteria can pass genetic information, whether it's transformation in the environment, transduction, between each other, and then the transformation, or excuse me, the conjugation using the sex pilus and like directly transferring, or excuse me, transformation is the bacteriophage and the conjugation is the sex pilus. And so anyways, all these genetic topics were never things I was interested in, but I found them really interesting, and the way you taught them was really, really beneficial. And so thank you for listening to my talk on epigenetics. I hope that was good enough. I'm sorry for wasting 36, and probably when I end this, probably 37 or 38 minutes of your life. I hope you get that back at some point, but I hope I made up for it at the end with the thank yous and the praise. So thanks, Dr. Ray. Have a great day, or night if you're watching this at night. I hope you learned a bit about epigenetics. I hope I did a good job explaining. So, yeah, cheers.